Hello students, this is Lohitha. Here we are into zoology class. So we are going to start learning a new chapter today, that is principles of inheritance and variation in today's class. Okay. So students, we have always been seeing that a mango tree is giving mangoes. A banana tree is always giving bananas only. A human being is giving birth to human beings only. Right. Dog is giving birth to dogs only all the time. Right. A cat gives birth to a cat all the time. Right. So this is why is this all happening? Why is a human being giving birth to a human being only? Why can't he give birth to a dog or a cat? Why is a banana tree giving bananas only? Why not mangoes? Why is mango tree always giving mangoes? Yes, so the reasons behind all these things will be dealt in the branch of science which is called as genetics. Okay, the genetics is a branch of science which is the study of inheritance and variation. Okay, it is the study of inheritance and variation. Okay, so these two words, inheritance and variations, we need to know very clearly about them. So first we will learn about what is inheritance. Inheritance is nothing but passing on the characters from one generation to another generation, from one generation to another generation is called as inheritance. Okay, inheritance is nothing but passing on the characters from one generation to another generation is called as inheritance. Okay, so students, some of the siblings appear same, right? The siblings, they appear somewhat similar, and the offsprings, we appear somewhat similar to our parents, right? Our parents, they appear somewhat similar to our grandparents. So that similarities between us and our siblings, we and our parents and our parents and our grandparents, these similarities are there, right? So those similarities are because of inheritance. That is nothing but inheritance, passing on the characters from one generation to another generation. So in some people, both the parents will be very tall and the children will be also very tall. So this tall character is actually inherited from the parents to the child. Okay, that means there is a passing of character related to height from the parent to child. So this is inheritance. Okay, and students, are we exactly similar to our parents? No. Are our parents exactly similar to our grandparents? No, right? So the differences in characters are nothing but variation. Okay, variation is nothing but the differences in characters. The differences in characters is nothing but variation. Okay, the differences in the characters is nothing but variation. We are not exactly similar to our parents. Our parents are not exactly similar to our grandparents. There are some differences. Though we show some resemblances, which is because of inheritance, but still we are showing some differences, that is variation. Okay, so this is the branch of science. Genetics is the branch of science which will be dealing with inheritance and variation only. Okay, so students, these two aspects, inheritance and variations, these are not new to humankind. Okay, so this inheritance of characters are known to humankind since around 8000 to 1000 BC only. 8000 BC to 1000 BC only, human beings, they have an idea about inheritance of characters. Okay, human beings during this time, 8000 to 1000 BC, human beings, they had a practice of selective breeding. Okay, they used to selectively breed and select the desirable characters from the offsprings. Okay, that was a practice which was there in BC's time. Okay, so they used to selectively breed the animals, and they used to select the desirable animals. Okay, one such wild cows are found in our India. They are called as Sahiwal cows, which are found in Punjab. These are the wild cows which are obtained due to this kind of selective breeding. Okay, so and in this in around 8000 to 1000 BC only, people also knew that this reasons for inheritance and variation of the characters lies in sexual reproduction. Okay, they also know that this inheritance and variation of characters, it lies in sexual reproduction. Due to reproduction only, this inheritance of characters and variation of the characters is seen because of sexual reproduction only. Okay, so here these people at those times, they used to selectively breed the animals and select the desirable characters from those animals. Okay, so this is about just an introduction. Over here, though people knew how to breed the organisms, though people knew how to select the desirable ones, they don't know what are the actual scientific basis for this inheritance and what are the basis for variation in the living organisms. Okay, so now later on, during 19th century, during the mid 19th century, the first experiments related to study of inheritance and variation were done. Okay, so let us learn about those experiments also. So students, we are now going to learn about Mendelian laws of inheritance. Mendelian laws of inheritance. Okay, so students, here you know this person who is called as Gregor Johann Mendel. Okay, Gregor Mendel. Okay, Gregor Mendel is the first person to perform some experiments related to inheritance of characters. Okay, so this was exactly in the mid 19th century, students. This Gregor Mendel is the first person to perform experiments related to inheritance and variation of the characters. Okay, so Gregor Mendel, he worked on pea plants, okay, whose scientific name is Pisum sativum. He worked on pea plant Pisum sativum for very long, 7 years, right, from 1856 to 1863. For 7 years he worked on this plant Pisum sativum. Okay, he worked on this pea plant Pisum sativum for very long, 7 years, and he proposed the laws of inheritance. Okay, he proposed the laws of inheritance. Okay, he proposed the laws of inheritance. Okay, so this person Mendel, he used to take large quantity of data. He used to give large quantity of data, which was a credit to his results. Okay, he used to take a large quantity of data, which was a benefit for him to acquire his results. Okay, so he has taken pure breeds of plants. Okay, he has bred pure breeds or true breeds or pure breeds. True breeds or pure breeds are those ones which have undergone self-fertilization for several generations. Okay, they have undergone self-fertilization for several generations and they show stable characters. Okay, that kind of breeds are called true breeds or pure breeds. They have undergone self-fertilization for several generations. They have undergone self-fertilization for several generations. Okay, true breeds or pure breeds are those ones which have undergone self-fertilization for several generations. These are those organisms which have undergone self-fertilization for several generations. Okay, and after that they show same stable characters. Okay, they are called as true breeds or pure breeds. Okay, they show same stable characters. They show same stable characters. Okay, they are called true breeds or pure breeds. So this person Mendel has selected all those true breeds or pure breeds, okay, for performing his crosses, okay. And this person Mendel, he has taken, because he has got pure breeds, right, so he has taken those plants which are different only in one character. Okay, he has taken the plants which are differing only in one character from each other. In such way he has selected the plants. Okay, so he has taken some seven types of characters. Okay, 7 types of characters, 14 types of plants. He has taken 7 types of characters which are shown by 14 different types of plants. Okay, because 7 types of characters shown by 14 types of 14 plants. So these characters are having alternate forms. Okay, why taking 14 plants? Because 7 types of characters, each of them having alternate forms. Each of them will have one alternate form. Okay, such kind of 7 characters he has taken, and he has taken 14 plants totally, each of which shows alternate characters. Okay, so in that way he has taken, and he has conducted several experiments on those plants considering those 7 characters. Okay, and he then proposed this Mendelian laws of inheritance. So let us see what are the characters that are taken by Mendel into consideration first. Characters and contrasting traits. Contrasting traits, as I have told you, they are appearing in 2 different forms, right? So he has taken how many characters into consideration? He has taken 7 characters into consideration. 1, 2, 3, 4, 5, 6 and 7. So he has taken 7 characters into consideration. So those 7 characters are plant height, okay, flower position, flower color, pod shape, pod color, seed shape and seed color. Okay, so these are the 7 characters that are taken into consideration by Mendel. As I have told you, each of them will be having 2 alternative forms. That means these 2 are contrasting traits. Contrasting traits, that means some kind of opposite traits, okay, opposite characters. So let us see what are the opposite characters or contrasting traits. So plant height, he has taken 2 contrasting characters, tall and dwarf. Okay, flower position, he has taken axial and terminal. Okay, flower color, he has taken violet and white. Violet and white. Pod shape, he has taken inflated and constricted. Inflated and constricted. Pod color, he has taken green and yellow. Seed shape, round and wrinkled. Round and wrinkled. Seed color, okay, yellow and green. So these are the 7 contrasting characters taken into consideration by Mendel. Okay, and what all the experiments performed by Mendel will be running around these 7 characters only. Okay, so he has taken the characters plant height, flower position, flower color, pod shape, pod color, seed shape, seed color. Okay, and what are those contrasting traits for each character? Let us see. Plant height, tall and dwarf. Flower position, axial and terminal. Flower color, white and violet. Pod shape, inflated or constricted. Pod color, green or yellow. Seed shape, round or wrinkled. Seed color, yellow or green. These are the characters which are taken into consideration by Mendel. Okay. So let us see next what are the crosses that he has made. Okay. So, students, one of the first characters that Mendel considered was the plant height. Okay. Plant height was a character that Mendel has first considered. So, let us see what did Mendel do, what were his observations, let us see now. Okay. So, Mendel, what he did first is he has taken the tall plants, pure bred tall plants, and he crossed it with the pure bred dwarf plants. Okay, pure bred tall plants were crossed with the pure bred dwarf plants. That means pure tall to the pure dwarf plants. Okay, so then what happened is in the offspring, so these are the parents. Okay, in the offspring, okay, of the first cross, the offspring of the first cross are said to be belonging to first filial generation. Okay, or simply F1 generation, first filial generation or F1 generation. This is something which we will be very frequently using, students, F1 generation or first filial generation. Okay, these are nothing but the offsprings of the first cross. So, tall plants were crossed with the dwarf plants. It resulted in tall plants only. It resulted in all tall plants only. Okay, all the plants which were obtained were tall only. Mendel was surprised with this. Okay, Mendel was surprised with seeing this all tall plants in spite of crossing the tall and dwarf. There are all tall plants only. Okay, so he has observed that this dwarf character is not appearing in the F1 generation. Okay, all of them were tall plants. All are tall plants. Okay. Then next what he did was he crossed this tall plants of the F1 generation with the other F1 plants. Okay, he crossed F1 tall plants to the F1 tall plants. Okay, F1 tall plants were crossed with the F1 tall plants. So here it resulted in F1 tall plants were crossed with the F1 tall plants. It resulted in 3/4 of the individuals, 3/4 of the plants are tall, 3/4 of the plants are tall, and only 1/4 of the plants are dwarf. 1/4 of the plants were dwarf. Okay, so 3/4 of the plants were tall and 1/4 of the plants were dwarf. Okay, so when a F1 tall plants were crossed with each other, it resulted in 3/4 tall plants and 1/4 dwarf plants. So here this is a cross performed between two F1 individuals, right? So the offspring here is said to be belonging to F2 generation or filial 2 generation, F2 generation or filial 2 generation. So in the F2 generation, 3/4 individuals obtained were tall and 1/4 individuals obtained were dwarf. Okay, so Mendel couldn't understand what could be the reason behind the tall only appearing in the F1 generation. Only all tall were there, but now which character which has not appeared in F1 has now appeared in the F2 generation. What could be the reason for this was a very big question mark for Mendel. Okay, so anyhow we will study how Mendel has solved this issue. Okay, so he observed that 3/4 of the tall plants and 1/4 of the dwarf plants were obtained as a cross between the two F1 tall plants. Okay, so here Mendel has made an observation that there is some factor, okay, there is some factor or there is something which is carrying this dwarf character from the F1 individuals to the F2 individuals. Okay, he has observed that there is something which is carrying this dwarf character from the parents to the F1, and from the F1 though it is not expressed, from the F1 it is carried to the F2 generation. So he called that particular thing which is carrying this character as a factor. Okay, he has observed that there are some factors, okay, there are some factors which are responsible for the height of the plants which are not expressed in the F1 generation, but they have expressed in the F2 generation. Okay, the reason why it happened he didn't know. Okay, so but he just simply said that there are something like factors which are carrying this character from the parents to the F2 generation, from the parents to the F2 generation. He called this as factors. These factors are actually now our present genes. These factors were then later on named as genes. Okay, these factors now later on they were named as genes. Okay, so these genes are those structures. These genes are actually the genetic material. These genes is actually the genetic material which denotes a particular character, which denotes a particular character, okay, which denotes a particular character, okay. So these genes appear in two alternate forms. These genes appear in two forms, okay. These genes appear in two forms. That means genetic material for every character is going to appear in two forms. The genes appear in two forms, okay. The genes appear in two forms which are called as alleles, okay. The genes appear in two forms which are called as alleles. The genes appear in two forms which are called as alleles, okay. Alleles are nothing, these two alleles are nothing but the two forms of the genes, okay. These alleles are nothing but the two forms of the genes. They show very minor differences between each other, okay. They show very minor differences between each other, okay. They are called as alleles, okay. So now he has got some clarity that there is some factor which is responsible for transmission of these characters, okay. So now later on what he did, let us learn. So students, the next thing is here, the reason behind, he tried to analyze the reason behind, what could be the reason behind having the tall character only completely in the F1 generation and having a tall and dwarf both in the F2 generation, okay. So the reason is here because some of the characters are appearing in the F1, but some of the characters are appearing both in F1 as well as F2. Some characters are not appearing in the F1 generation. So it was thought that those characters, okay, which are not appearing in the F1 generation may be recessive characters, okay. Those characters appearing in F1 which are not appearing, those characters which are not appearing in the F1 generation are called as recessive characters, okay. Those characters which are not appearing in the F1 generation are called as recessive characters, okay. Those characters appearing in both F1 and F2 generations are called as dominant characters, okay. They are called as dominant characters, okay. Those characters which are appearing in F1 generation are called as dominant characters. Those characters appearing only in F1 and F2 are called as dominant characters. Those appearing only in the which are appearing only in the F2 but not appearing in the F1 generation are called as recessive characters, okay. So dominant means appear in both generations. Recessive means they don't appear in any other generations, they appear in the only second generation. In first generation they are not appearing, okay. So how can we say what are the alleles which are defining which characters, what character is dominant, what character is recessive? To denote that, we can use some symbols, okay. To denote that we can use some symbols. Let us see what those symbols that we can use, okay. So before that we will need to learn about few more terms. Phenotype. Phenotype is nothing but the appearance of the organism, the appearance of the organism. Whatever the characters that we can observe from an organism externally, that is phenotype, okay. Whatever the characters that we can observe from externally is called as phenotype, okay. The next is genotype. Genotype is nothing but the genetic makeup of the organism. The genetic makeup of the organism is called as genotype. Okay, genotype is the genetic makeup of the organism. Okay, so students, we use alphabets for representing the alleles. So we are using, for a tall allele we are using capital T for representing the tall allele. For a dwarf allele we are using small t. Here we can use a T and a D also. Okay, here we can use T and D also, but these characters, these alleles, they may help us in confusing about the different characters. Okay, we can, if there is a D, we can think of anything about a D, right? So for that reason, to denote very clearly if what is dominant, what is recessive, what is the character that they are expressing, we are using capital letters and small letters for denoting the characters. We are not using any other alphabets. We are using the same alphabet in the upper case and in the lower case. In that way we are using the alphabets to represent regarding the characters. Okay, T, capital alphabet, we are using it for denoting the tall character. Small alphabet we are using it for denoting the recessive character. Okay, so students, here the tall individual is crossed to a dwarf plant, right? Tall is crossed to a dwarf plant. So this tall and dwarf characters, these are the phenotypes. Okay, these are the phenotypes. Okay, and what are the alleles that they may contain is the tall individual, because they are purebred, they contain TT. Okay, 2 tall alleles they are having. Dwarf allele, because they are also purebred, they have 2 small t's. So now, because this is the genotype, here we are representing the genetic makeup, genotype. These are having 2 dominant alleles, these are having 2 recessive alleles. Okay. So now from these gametes are produced. So always we represent gametes in the circles. Inside the circles we represent the gametes. So these 2 gametes, they will fuse together, because there are only 2 same alphabets, capital T, small t. One T is enough for us to represent the gamete. Okay. So capital T, small t, when they are undergoing fertilization, it will result in the F1 generation, which is going to be capital T, small t. And what did Mendel observe in the F1 generation? All the trees were tall only. All the T's were tall only. So this capital T, small t is also a tall character. Okay. So because here the tall allele T is expressing here, and the small allele, small t, which is the dwarf allele, is not expressing, we can understand that this capital T is dominant allele, capital T is a dominant allele, and small t is a recessive allele. We can understand that the small t is a recessive allele. Okay, this is what we can understand from these alleles. And you know students, one more thing that we need to learn from this thing is, here you can observe 2 capital T's being tall, right? And one capital T, one small t is also being tall. Yes. So here and we can observe that here 2 small t's are present. So if there are 2 same alleles, okay, if there are 2 same alleles, then that condition is said to be homozygous. Okay, that condition is said to be homozygous. If the 2 alleles are same, then that condition is said to be homozygous. Okay. If the 2 alleles are different, like in case of this F1 individual, okay, we call this to be heterozygous. The parent plants, they are homozygous, you can see, and the F1 generation individual is a heterozygous individual. Okay. So students, we need to learn few more things. So here we are representing the the plants which are different in only one character. Only one character is there is only one character difference. Okay, there is only one character difference between the two crossed plants, right? So there is a tall plant and a dwarf plant. Otherwise all the other characters are same in the plants. Okay, so whatever the plant we have obtained in the earlier cross, capital T cross small t, we have obtained a capital T small t, right? So this plant is a heterozygote. This plant is a heterozygote. Okay, this plant is a heterozygote. So this plant, whatever crossed in this here, they are heterozygote only to one character. Okay, they are heterozygote only to one character. So this cross, this individual whatever obtained is here is called as a monohybrid. Okay, here this individual whatever formed is here called as monohybrid. Monohybrid in the sense it is having difference in only one character. Only one character is difference between the other plants. So it is called as a monohybrid. And this cross whatever is performed here is called as monohybrid cross. That means the cross in which we are considering only one contrasting trait, that is called as a monohybrid cross. The cross in which we are considering only one contrasting trait, it is called as monohybrid cross. Okay. And students, the results of this crossing can be very clearly understood by drawing Punnett squares. By drawing Punnett squares. Okay, these Punnett squares were designed by Reginald C. Punnett. Okay, Reginald C. Punnett has designed these Punnett squares which help us in very clearly understanding about the crosses. Okay, let us perform this let us draw Punnett square even for our cross. Okay, so before we go for Punnett square, we need to learn few more things, students, because this is all just basics. Okay. So always this symbol is used to indicate female. Okay, in a Punnett square the symbol is used to indicate a female and this symbol is used to indicate a male. Okay, it is used to indicate a male. So here from this Punnett square we use the possible type of gametes. The possible type of gametes from both males and females. From both males and females. Okay, we take the possible type of gametes from both males and females. Okay, so students, and we use the possible type of gametes from both males and females, and we derive the possible genotypes and the phenotypes of the offsprings Okay, we derive the possible genotypes and phenotypes of the offsprings. Okay, so first let us draw a Punnett square for this cross. Capital T cross to small t. That means a tall plant is crossed to a dwarf plant. So what are the type of gametes produced, students? So this is a phenotype, this is the genotype. Let us see the gametes, what are they produced, gametes. So tall TT, both are same, no? So only one type of gametes, capital T. Dwarf, capital T, small t, no? So small t, these are the type of gametes produced by the small t. So now let us draw a cross between these two. So here we are going to draw a Punnett square. Okay. So here is going to be the female. However, you can write female I have written here and this side male I have written. Okay. So the possible type of gametes can be both. Okay. So this can be capital T and small t. It can be from the male, from the female, capital T, small t. Okay. The ova can be carrying the capital T or small t, or sorry, the egg can be carrying the capital T, small t, or even the pollen can carry the capital T or small t. So these are the possible type of gametes. Okay. So now we cross capital T in this way. Okay. I'll just put the dotted line, students, because this is the first time we are doing this. Okay. Capital T, capital T, small t, capital T, small t. Okay. Once again, now after we are done with this, we did the cross with the capital T small t and capital T small t. Okay. So now we are done with this. We have to go with the next one. So this small t, capital T small t. Again this t with this t, small t small t. Okay. It should be actually, possible type of gametes are only two, right? So T and T, these are the type of gametes that we need to write, students. So female, male, okay, because this is the first generation we are doing. Okay. So capital T, small t, capital T small t, capital T small t, capital T small t, capital T small t. So this capital T small t, all of them were tall only, all of them were tall only. So here we have observed what are the possible type of gametes, and we have also observed what are the possible genotypes and what are the possible phenotypes. All the four individuals, they were tall only. Okay. Now let us see what happens in the F1 generation, F2 generation. So what is the F1 generation individual? F1 generation individuals were capital T small t. So now this is F1 generation individual. If we are crossing two F1 individuals, capital T small t to a capital T small t, all of which are tall only, we get two type of gametes, capital T small t, capital T small t. Okay, four type of gametes we have got. So we are going to draw a Punnett square that we have actually drawn earlier. Okay. So this side we indicate the female, this side we are indicating male, capital T small t, capital T small t. This capital T with this capital T, capital T, capital T. This capital T with this small t, capital T small t. Coming to this small t, capital T, small capital T small t. Small t, small t, small t, small t. So this individual is going to be, this individual was found to be tall, this individual was also found to be tall, this is also tall, but this was dwarf. So from this observation we have observed that 3/4 of the trees were tall and only 1/4 of the trees were dwarf. Okay, 3/4 of the trees were tall and 1/4 of the trees were dwarf. So here from the phenotypic ratio of this cross, this monohybrid cross will be 3:1, because 3 tall, 1 dwarf. So this ratio will be 3:1. Okay. So 3/4 is to 1/4. 3/4 is to 1/4, which is nothing but 3:1. Okay. Coming to the genotypic ratio. Okay. The genotypic ratio would be 1:4, which is capital T, out of 4 only 1 was capital T, right? And 1:2, that means only half, that means out of 4, 2 were capital T small t's, and 1:4 small t. Okay. So this means this is like in the form of a plus b whole square formula, Okay. So it is going to follow the mathematical formula a plus b whole square. Okay. It is like 1/4 plus 1/4 whole square. Okay. It is like a plus b whole square. This genotypic ratio is like a plus b whole square. So if we can deduce this ratio, we get 1:2:1. Okay. Which is 1 capital T, 2 capital T small t, and 2 small t's. Okay. This will be the genotypic ratio. Okay. So students, this is how we obtain the genotypic ratio and phenotypic ratios from the Punnett square. By using Punnett square, this is how we obtain the phenotypic and genotypic ratios. Okay. The next thing we are going to learn is here, the next thing that we will be learning is how do we understand if what is the phenotypic ratio and genotypic ratio of the plant. Okay, how we can identify what is the phenotypic ratio, genotypic ratio means by the Punnett square. But how can we identify if the plant, particular plant F1 generation individual is a homozygote or a heterozygote. Okay, how can we identify if a F1 individual is a homozygote or F1 individual is a heterozygote. Okay, so students, if we have to identify a plant is homozygote or heterozygote dwarf. Okay, if we have to identify if a particular plant is a homozygote or heterozygote dwarf, then what do? Is we just cross this heterozygote, whatever dwarf individual is there, we continuously go for crossing of the dwarf individuals, self crossing of the dwarf individuals, of the F1 individuals, of the F2 individuals, F3 individuals. In that way, this dwarf, when they were crossed to dwarf, even in the F3, even in the F3 and F4 generations, they were found to be dwarf only. Even in the F3 and F4 generations, they were found to be dwarf only. With that we can understand that, this character, because even in the successive crossings also it is showing the dwarf character only, we can understand that these individuals, they are homozygote dwarfs. These individuals, they are homozygote dwarfs. Okay, in that way we can identify the individual if it is a homozygote dwarf or heterozygote dwarf. When coming to tall, how can we identify? That we will be learning in the next class, students. Okay, so we need to learn few more things related to the same concept, monohybrid cross. We will learn regarding the next concepts in monohybrid cross in the next class. So this is all for today, students. Thank you all for joining the class. We'll meet tomorrow. Thank you all.